and so forth. But in each case, we kind of have an idea of what's going on. So if you take cancer, for instance, horrible as different cancers are, one has a good understanding of what's going on. Okay, so we have DNA in the nuclei of the cells. If the DNA is damaged, you need radiation, melanoma, or improper copying, or whatever the case may be, mutagens, so forth. Okay, the DNA will code for improper proteins. Okay, and if those improper proteins are important for maintaining a cell cycle, okay, you lose control over the cell cycle, you start building up cellular masses, and that's where the tumor is. Okay, so that opens up lots of avenues for being able to treat cancers. Number one is you can isolate, if possible, you can isolate the tumor and remove it. There's chemotherapy, there's radiation therapy, there are drugs that will act at receptors on tumor masses. Okay, and similar stories with other types of diseases. With the neurodegenerative diseases and the way they spread through protein aggregation, there's no sense whatsoever as to how to attack these diseases. So number one, we don't even know really what propagates the infectious form, okay, or the improperly folded form. So here we have single proteins, they form small little aggregates, they come together into larger aggregates, they form fibrils. It's not really even clear among this family of all these different structures, okay, of proteins, which ones are really toxic, okay? So where do you even begin to design a drug that interferes with this process? And in fact, some of the drugs, they don't make it into the clinic, thank heaven, but some of the drugs that are tried at the cellular and animal level actually exacerbate the neurodegeneration. They don't make it better because they actually end up arresting this process at a place where some of the proteins actually retain a higher level of toxicity, okay? So the problem with neurodegenerative disease, and why it's, as I said in the title, it's so damn hard to cure Alzheimer's disease, is these protein plaques, okay, that interfere with cellular function are fully interdigitated through all the cells in the vicinity of the plaques. So there's no way to go in and cut out an Alzheimer's plaque. You're necessarily going to be removing neurons as well. If you want to design a drug, okay, that interferes with this process, yeah, if you could stop the whole thing, that's great. A lot of scientists have focused on halting this over here, but it's very, very